Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Trying to figure out what's really going on with a teenager deep down, it can sometimes feel, well, impossible, especially if they're struggling inside. Maybe they don't have the words or, they just don't feel comfortable opening up.
Right. And the way we currently diagnose something like adolescent depression, it leans so heavily on them telling us their symptoms.
Which can be really tough for them, It might not be the whole story.
Exactly. It's a real challenge. That reliance on subjective reports, it means we might miss things or maybe delay help when getting in early could make such a difference.
Totally. And that's why this next deep dive really grabbed our attention. We're looking at some pretty fascinating research exploring a totally different angle using a simple blood test. A blood test to potentially help identify depression in teenagers.
Wow, okay. So moving towards something more objective, a biological signal.
Precisely. It's aiming to find something measurable beyond just asking, how do you feel? This work comes mainly from McGill University. It was published in Biological Psychiatry, Global Open Science, and a health news article picked up on it too.
Ah, okay. So we get the detailed science and also kind of the broader take on what it could mean.
Yeah. That's the plan. Our mission today is to really unpack this study. We want to understand, the idea behind using blood tests for this.
How they actually did it.
Exactly. How they did it, what they found, and maybe the biggest question, what could this all mean down the line for helping teens who are dealing with depression?
Sounds good. Let's dive in.
Okay, so let's unpack the study itself. The researchers at heart, they wanted to find specific biological signals, markers in the blood.
Right.
Markers linked to depression, but specifically in teenagers. They suspected that teen depression might look different biologically than adult depression.
So they were hunting for something potentially unique to that stage of life.
That seems to be the idea, looking for markers distinct to adolescence.
And the specific markers they zeroed in on are called microRNAs.
microRNAs. Okay. Can you break that down a bit? What are microRNAs?
Sure. So think of them as really tiny molecules inside our cells. They're involved in genetics, but they don't, carry the main instructions for building proteins.
Okay.
They act more like volume controls or maybe dimmer switches for our genes. They help control if a gene makes a protein and how much it makes.
So they're regulating gene expression.
That's it. They're regulators. And crucially, for this study anyway, they circulate in our bloodstream.
Ah, so you can find them easily.
Relatively easily, yes.
Yeah.
Makes them accessible for testing.
That accessibility feels key. So how did they actually get the samples from the teens in the study? Was it a big blood draw?
No, this is where it gets really practical. They had 62 teenagers involved, 34 with a depression diagnosis, 28 without.
Right.
And instead of a standard needle in the vein draw, they used dried blood spots.
Dried blood spots, you mean like a finger prick?
Exactly that. Just a simple finger prick, collected a few drops of blood on a special card, let it dry, done.
Wow, that's much less invasive. I imagine that's way easier, especially with teenagers.
Oh, absolutely.
Yeah.
The sources really emphasize this. It's minimally invasive, and those dried cards, super easy to store, easy to transport.
Which is great for research, right?
Yeah.
Makes bigger studies more feasible.
Definitely. Could be essential if this ever scales up.
And didn't the McGill team have to develop a new lab method for this to get those microRNAs out of those tiny dried spots?
Yes, that's right. They needed a novel way to efficiently extract and then analyze these tiny molecules from such small, dried samples.
And how did they analyze them? What was the tech?
They used a technique called small ribonucleic acid sequencing.
Small ribonucleic acid sequencing. Okay, what does that do?
It's basically a really advanced method to read the signature of all the different microRNAs present in that blood spot.
So it tells you which ones are there and how much of each?
Exactly. It gives you a whole profile.
Okay, so they get the spots, use the special method, profile the microRNAs. What was the first big thing they found?
Well, the first major finding was identifying 9 specific microRNAs.
9?
9. And the levels of these 9 were significantly higher, elevated in the blood of the teenagers who had been diagnosed with depression, compared to the control group.
So a distinct pattern linked to the diagnosis in these young people.
That's what they saw, yes. A clear correlation in this group.
Now, here's where I thought it got really interesting. The sources stress that these specific 9 microRNAs, they haven't been linked to depression in adults before.
Right. And that's a critical point. It strongly suggests these molecules might be tied into biological processes that are, unique to the adolescent brain while it's still developing.
Which could be part of why depression can manifest so differently during those years.
That's the implication. Finding unique biological markers really supports the idea that the underlying biology might differ between teen and adult depression.
That makes a lot of sense. Okay, so identifying the markers was step one. Did the study go further? Like, could these markers predict anything else?
Yes, and this is where some really exciting, though still early, findings came in. They did some exploratory analysis
Okay.
and found that the levels of 3 of those 9 microRNAs, let me get the names right, microRNA 36135P, microRNA 30C2, and microRNA 9425P.
Right?
Their levels in the initial blood sample seemed to correlate with how severe the teenagers' depression symptoms were when they were followed up months later.
Wow. So higher levels of those three predicted more severe symptoms down the road.
That's what this initial analysis suggested. And importantly, they checked this against anxiety symptoms too.
Oh.
And the link was specific to depression severity, not anxiety. That points towards a connection with the actual depression process itself.
That specificity is really interesting. It makes you wonder, could a test like this potentially flag teens who might be at risk for more severe or maybe more persistent depression earlier?
That's the hope, isn't it? Getting ahead of it before symptoms become really entrenched and maybe harder to treat.
Yeah.
The senior author, Cecilia Flores, she actually spoke to this point. She mentioned how alarming the rise in adolescent depression is and how early onset can have these really severe, long-lasting effects.
Like substance use, isolation.
Exactly. And difficulties responding to treatment. She highlighted that finding markers like these could potentially help before things get that tough.
So identify risk earlier. That would be huge. Did they also look at where these microRNAs are active in the body, like in the brain?
They did. They used existing genetic databases to see where these microRNAs are typically expressed.
Yeah.
And they found them expressed in key brain regions we already know are involved in mood disorders. Places like the hippocampus.
Memory and emotion.
Right. And the substantia nigra, parts of the frontal lobe which handles executive functions, mood regulation.
So they're showing up in the right neighborhoods, biologically speaking.
Pretty much. And they even found a direct link, though again, exploratory, between one microRNA and a brain structure.
Which one was that?
Higher levels of microRNA 32.5P were associated with a smaller volume in the hippocampus in the teens they scanned.
Smaller hippocampus. That's something often seen in depression research, isn't it?
It is. And they found this link even after adjusting for things like overall brain size or whether the teen was taking medication.
So it's not proof of cause and effect, but it's another strong correlation, a physical link between the blood marker and the brain.
Exactly. It points towards a potential mechanism. And they went even further, using computational tools to predict which genes these microRNAs might be controlling.
Ah, back to the dimmer switch idea.
Right. The analysis suggested these microRNAs target genes involved in really crucial stuff. Brain development, how neurons talk to each other, information processing.
And pathways already linked to depression?
Yes. They gave examples like a gene called EPAK7, involved in neuromaturation, another ERBB4, which has links to adult depression, and BCL11B, critical for brain development generally.
So it suggests these aren't just passive bystanders. They might be actively involved in the biology driving the depression.
That's the hypothesis these findings generate, yes.
Okay, let's circle back to the practical side for a moment. That dried blood spot method,
Yeah.
it sounds like a real, game changer for doing this kind of research.
It really seems to be. The first author, Alice Morganova, called it a practical tool for psychiatric research.
Because it's minimally invasive.
Right. Allows tracking of these early biological changes without a major procedure. Makes it easier on participants, easier for researchers logistically.
You could potentially do studies outside of big hospital clinics, maybe reach more diverse groups of people.
Exactly. It opens doors for future research and maybe eventually broader uses like screening, although that's still a way off.
So, okay, the big picture then. What does this all add up to? It feels like this research offers a really exciting glimpse into a possible biological basis for teen depression.
And one that might be unique to that age group.
Yeah. Suggesting we could potentially use objective markers from a simple blood test to help out alongside the current ways we diagnose, which relies so much on self-report.
It definitely hints at that potential. Maybe earlier detection, maybe identifying kids at higher risk.
And maybe even guiding treatment, more tailored interventions based on someone's specific biological profile.
That's the long-term hope, yes. moving beyond a one-size-fits-all approach.
But, and this is a big but, we need to be clear, right? The sources emphasize this is discovery phase research.
Absolutely crucial point. This is foundational work. It's a starting point, not the finish line. The findings are promising, definitely, but they need a lot more validation.
Like those findings about predicting future symptom severity or the link with the hippocampus size.
Exactly. Those came from exploratory analyses based on smaller numbers of participants who had that specific follow-up data or the brain scans.
So they need to be repeated.
In much larger independent groups of teenagers.
Yeah.
To make sure the findings are solid and really do apply more broadly.
And what about using it as a diagnostic test right now? Did they look at that?
They did look at whether the microRNAs could predict a diagnosis at the time of the test. 2 markers showed some potential, but the accuracy just wasn't there yet. The analysis showed real limitations.
Meaning too many false positives or false negatives?
Essentially, yes. The stats they reported suggest that if you used it as a standalone test now, you'd risk misdiagnosing people, either flagging someone who isn't depressed or missing someone who is.
So definitely not ready for clinic use as a diagnostic tool on its own.
Not even close, based on this data. It shows an association, but not the accuracy needed for diagnosis. Needs much more work.
Okay. So lots more research needed there. And also, presumably, research into how these microRNAs actually work, not just finding links, but figuring out the mechanisms.
Precisely. We need studies that dig into how these molecules might actively influence brain development and function during adolescence to contribute to depression.
And looking at how genetics and environment play into this whole picture too, I imagine.
For sure. Future studies need to confirm these initial findings in big, independent samples and also explore those complex interactions.
But this study gives them a place to start, right? A framework.
Absolutely. It gives researchers these specific microRNA targets to investigate further, focusing the search.
Okay. So, to sum it all up, this McGill research is, well, it's genuinely exciting. It points towards a potential new avenue using objective biological signals in blood to better understand and maybe intervene earlier in adolescent depression.
It's early days, as we've said, and the researchers are rightly cautious. But it lays really important groundwork for digging into the specific biology of depression in teenagers, which seems increasingly distinct from adult depression.
Which leaves us, and you, with a final thought to chew on. As we keep learning more about the unique biological shifts happening during that really critical time of adolescence, how might our whole approach need to change? How we understand, how we diagnose, and how we support young people's mental health? How might that evolve in the years ahead?
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.